Hi, welcome back to the Leukopak Lady. Today, I'm giving you a recap of my experience at the 3rd Annual Donor Selection and Cell Source Summit hosted by Hanson Wade. So this was a small conference, but many of the right people were in the room. There were 11 different material providers represented, 4 of which were conference sponsors. The developers attending range from academics to cell therapy company scientists and executives. A major topic covered was how to identify the best material and donors. Some use cord blood to manufacture their therapies, while many others use immune cells from buffy coats or leukopaks. The speakers covered their approaches and experiences of finding those best donors or identifying the best material. One great point made was that the screening process needs to look like the manufacturing process. There was also agreement that AI and machine learning tools should be employed where possible to help. Some speakers covered how to activate or stimulate the best cellular function during manufacturing. Outside of the box things like donor cannabis use, bacterial extracts, and intact bacteria were discussed. There was also a great conversation around the term superdonor, which is not well defined. Some advocated for this term to not be used because it can't be defined universally, while others loved the term and stressed for the industry to adopt it and put it on blast. It's true that the definition of a superdonor will need to be different for each developer, each therapy, and each indication, but I would love to see some basic specifications defined. I'm hoping to see laser force cytology and flow platforms like Excelix be used more widely to identify superdonors. Another interesting approach brought up during the roundtable discussion was, instead of defining ideal donor and material specifications, developers should instead adjust their manufacturing processes to account for material variability. Now, I personally strongly disagree with this tactic, but I learned something from the discussion around it. And I'm anxious to see clinical data from companies using this approach because maybe I will change my mind. One suggested that developers are solely responsible for defining their ideal donor and material specifications, and that the collectors should play little to no role in this process. And while I agree that developers should define these for themselves, I also think that collectors can and should play a role in sorting out the bad donors so that the likelihood of finding a super donor within a given donor pool is much higher. Next, I asked many attendees what they liked about the conference or what their main take-home message was. Listen to their responses.
The best part is that we have suppliers and customers in one room, so we can really talk to each other about what's important to us, what you guys need to know from us as a supplier.
And I've been working in the lab primarily, and I've learned a lot about the nuance of the other side of the collection, and just seeing how much work goes into that, and how it just gives me a different perspective of what we can expect from our collection agencies. So yeah, I'm getting more perspective.
Yeah, great.
This time I got the chance to meet people who actually source the donors, and everyone who's interested in understanding donor variability was in this conference.
I think that the quality of the conferences and the attendees has been outstanding.
Yeah, and I really have liked that dialogue between the developers and the collectors, and having conversations about ideal donors or super donors and donor screening and learning from each other. So I really enjoyed that myself.
Yeah.
I love how collaborative it is. It's really nice to kind of just be able to bounce your questions and ideas and open lines of communication.
I think it's always informative. You get a lot of different perspectives from across the industry. You get to hear what other collection providers are doing and talking about, and then you also hear from the manufacturers and the developers to piece all this together.
I also feel like in the conference, the network that you're building, all the connections, everyone is working on this niche industry, and so it's great to meet everyone and build those relationships.
And I like that questions that came up last year at the conference have been somewhat addressed this year.
That's great.
So I think that there's really good ideas that people are sharing and real life outcomes and stuff that we're seeing from everything that we've been talking about the last couple of years.
Thanks for joining me for this conference recap. Next week, I'm going to post my entire interview with Tracy Ryan, where she talks in detail about the use of cannabis and cannabinoid receptor stimulation to supercharge NK cells to fight cancer. It's like nothing you've ever heard before.